So, good morning, everyone, and welcome to the second talk on the Neuroscience Thematic Week. Today with us, we have Dr. Liliana Pedro. She is Portuguese, and she came in Germany in 2015 to study neuroscience and how different brain cells develop and how they work together to keep each other healthy. So last year, she finished her studies, and she's now working for a company that is researching new medications and new therapies for different neurodegenerative diseases. She's going to tell us about it. Type of these diseases are Alzheimer's, Parkinson, and dementia. So now she's based in Hamburg, and she's going to talk to us about how we can use our skin cells to study our brain. The floor is yours, Liliana.
Hi, hello everyone. If you don't mind, I would like you to help me with this talk, and I would like us to have a conversation about it. So feel free to either write on the chat or speak through the microphone and just have a discussion with me. And I do want to talk to you about how we can use your skin cells to study your brain. And actually, I am doing that in my job, and it's something that will revolutionize the way not only that we do new medications, but also in the way that we understand how the brain works and how different diseases affect how the brain works. Wait, did we lose them?
It could be a connection issue. Don't worry, they will reconnect if it is a problem. But I suggest we can go ahead so that we have the recording full and someone can follow.
Okay. So then I want to start by showing you, for example, what a human brain looks like. So this is basically the shape of a human brain if you see it from this side, with this part being the front side and this part here being the back of your skull. And your brain is basically the size that if you hold your two hands like this is about the size of your brain. And an adult brain weighs about 1.5 kilos. So take a bag of rice, put that in your hands, and you will have an idea about how much your brain weighs actually. And even though this is what we see of our brain, if we look deeper into it and we use a microscope so that we can look at smaller, very tiny things, you will be able to see cells. Cells, in this case, brain cells. These usually are the cells that are most commonly known within the brain. They are not the only ones, but as the most famous ones. And these brain cells or neurons then work together so that they can create not only the brain structure, but also the function. So it allows you to think, allows you to pass along information, allows you to move. These brain cells, even though here is a little cartoon, if you again look under the microscope, we can use different colors so that we can see them better. And this is how they look like. So you have this part here in the middle, which is the center or the body, and you can see here. And then you have all these little feet and this long one. All of these little structures around the head and the ones at the end of the feet, all of these little structures are used so that these cells can communicate with each other, which is then what you can see here in these green dots. This is what they use to communicate. So you can think if you touch your colleagues on the shoulder and your other colleague on your next side on the shoulder, and the first person thinks of a number, by tapping on the shoulder of the next person, you could pass along which number the first person thought about. And you could talk just by using the simple technique. And the brain cells, even though they don't use tapping, it's the same idea. So they use the little feet on one side and the little heads on the other side, and they pass information from the feet to the head. And this is how they look like inside your brain. Of course, even though all of our organs, our brains, our livers, our hearts, our muscles, all of them are made of different cell types. So brains have neuronal cells, livers have liver cells, skins have skin cells. All of them are made from cells. But if you think back to your biology class about how everything started, we humans develop from a single cell, because you have the egg from the mother and you have the sperm from the father. And when they combine, they form what is called a zygote. And this is one single cell. This one single cell then divides and divides and divides and divides and divides until later on you can form a full being that has a brain, that has a heart, that has a liver. These cells that you start from the beginning that can then originate every single cell and every single organ in your body, these are called stem cells. Hi. These are called stem cells. These are cells that can create any single cell in your body. And this is important. Why? Because this is exactly where I work on. So you have stem cells, and they are called stem cells, or more exactly, pluripotent stem cells, because they indeed can form brain cells, they can form skin cells, they can form blood cells, and all the cells in your body. Interestingly, if we want to make a stem cell into a brain cell, which I do a lot in my lab, this is what it looks like under a microscope. So this is what the stem cells look like. And I want you to pay attention to this little guy here. So this is what it looks like. It's not as round as in the picture because they are attaching on the bottom. And even though we think of cells as one thing that doesn't move, it's actually not true. So what you will see is that if you pay attention to this little cell here, you'll see that it uses its little feet to move from here all the way to here. So if you pay attention to that little cell and here, you can see how it moves along by crawling around, right? And as it crawls around, if you pay attention here, this shape is quite different. This shape is very different. And as the movie or the video goes along, you can pay attention to the cells and you can see that they change their shape. They go from this little simple shape that is more roundish to a shape that has a more elongated structure. So let's take a look. So they go around and then you see how they start changing shapes and they become more elongated. As they become more elongated, they start moving less and less and less. And now we have these structures that they don't move that much, but they have all of these little wires that go along. These little wires, we can think of them as exactly the wiring of your brain. This is exactly how your brain communicates. It extends these little structures, these little wires, so these little long feet that you can see here, so that it can communicate with the next cell. And this is exactly what we see under the microscope when we use stem cells to create brain cells. So I will pass the movie along so that you can see again. So they start as very round, they move a lot, and then they start slowing down, changing their structure, becoming with more little feet and more little feet and more little feet, until they form what we call a network structure. And this is what we use in the lab to study the brain. Of course, stem cells, if you want to get them naturally, they would come either from when an egg joins with a sperm and they form the little zygote, or they can come from the umbilical cord of babies. And that's why many parents, once the babies are born, choose to save the cells that are inside the umbilical cord. And for many years, this was the only way that we could get stem cells. This was the only way that we could study this process in which stem cells can become any cell. But as you can see, this is not so easy to get, and it's very difficult and it's very expensive. So this is not something that we would use a lot. Also, it's a very limited thing because the umbilical cord only has a certain number of cells. They might not be healthy, so we might not be able to use them. So you don't have that many that you can work on in a lab where we use millions of cells at a time. So in 2012, so not that long ago, if you think in terms of science time, a person came up with a solution for this in which we can make a stem cell out of a skin cell. And how does this happen? Stem cells, usually they only become another cell. They become a neuron, they become a liver. It doesn't really work out the other way around. A neuron cannot become a stem cell. A skin cell cannot become a stem cell. And this is because when we age, we are not going to go back to baby form. We always get older. So it is very unnatural and not normal to make a skin cell into a stem cell. And this can only be done by a process that is called reprogramming. This process basically is forcing, by brute force, the skin cell to forget that it's a skin cell. It forces it to become a stem cell, to become a baby cell. And this is a very harsh process. And because these stem cells are no longer exactly the same as the ones that you would get from an umbilical cord, we needed to find a way to distinguish them when we use them in the lab. So these cells, these stem cells, they're originated from a skin cell or a muscle cell. They are called then induced. They are induced because they were forced to become a stem cell. And these cells then will behave exactly the same as an umbilical cord stem cell. And we then in the lab can make them into a neuronal cell, a skin cell, and a blood cell. And this process then is what we use in the lab. So these days, it's very, very easy to get stem cells to study any type of disease, to study any type of organ, because we can just take them from the skin, which is very easily accessible. It's right here. So it's very easy to take some skin sample from a person and make it into a stem cell that we can study everything from the person. But again, this is not a natural process, but it makes the scientist's life quite easy, because this means that from a skin cell, we can get a brain. And what I mean by this is that we could potentially take a skin cell from any of us by using this reprogramming method, force it to become a stem cell. This stem cell, then, we can choose what we want it to be. In my case, because I study brain diseases, I make it into a brain cell, which then I can actually, in the lab nowadays, we can make what is called mini brains. Of course, these are mini brains that I'm talking about. They are not exactly human brains, and they will never become a human brain in the size or even in the complexity, but they are enough that we can study how certain diseases develop. What do these mini brains look like in reality? They look like these little balls. This is exactly how they look like. So this is what we call a Petri dish, and there is a solution on it that this is basically keeping the cells happy, like our blood is maintaining the cells. The cells don't exist in a dry place. So these are little balls of brain cells that we just make in the lab. And then this is what they look like. And over time, these little balls can grow and grow and grow to a limit, which is about 3 to 4 millimeters in size. You can see them here in this video, how they grow and become a little bit bigger. And of course, this is their limit, because after this size, they are too big and we can no longer keep them in the lab and they will die. But by this size, they have developed different structures, and they even have developed the capacity that the different brain cells can communicate with each other exactly the same manner that our brain communicates, which is this picture here. So this is one of these balls looked under the microscope. And then what you will see is that when a brain cell communicates with another brain cell, it releases an electrical energy. And this is what you're seeing here. So when it lights up, it's a cell communicating with another. And you can see that this actually happens quite fast. And there's entire networks that connect this side and then this side, and it just goes around and is communicating with each other. And then this, for example, we use in the lab that we can test different medications and different medications and different drugs, so that, for example, we can improve brain communication, or we can make it worse, for example, when we test poisons. And we can use all of these tiny brains to study not only the normal way that a brain would develop and that the brain would connect and that the brain would communicate, but we can also test, for example, if you have a disease, how does that disease influence the way that the brain communicates, the way that the brain develops? And this is exactly why these stem cells are so important, because this would never be possible if we didn't have a large access to them, if we didn't have access to stem cells that come from patients. Because This then is the part where if we have a patient that has a disease, let's imagine, for example, a patient with Parkinson's disease, which is a brain disease that affects people, and when they develop this disease, these people have parts of their brain that will die out, that will die, and then these patients will develop what we call tremors, so they shake a lot, and this makes it very hard for them to walk and to eat because it's very hard to lift a spoon, and they need a lot of help. So if we have a patient, for example, a Parkinson's patient, we can take this patient, and then by doing a biopsy, which means taking a little bit of their skin, we can take some skin cells from the patient. Then these skin cells, we can use a technique to make them stem cells that it's called reprogramming. By using this reprogramming, we can create a stem cell that is specific for this patient. The stem cell, like the skin cell, belong to the patient, and they will behave the same as the patient. So by having this patient-specific stem cell, we can make it into a brain cell because this patient has a brain disease. So it's of interest to us to have a brain disease patient cell. With this, we can test different drugs that exist in the market and see whether or not this patient reacts in a good way to these drugs. So we can test thousands of drugs just by using these patient cells. And then by using these drugs, if they work on the patient cells, then it's much more likely that they will be able to work on the patient. And so we can treat the patient successfully with these medications or with these drugs. This is exactly what I do in my job, for example. There are other options if the patient has, for example, a genetic disease. These are diseases where the patient cells, for some reason, don't work properly because something is broken inside of them and they cannot function correctly. In these cases, we can do something where we can change the genes so that you will have a healthy stem cell. These healthy stem cells, then we can make healthy brain cells, which we can transplant into the patient because these patients, these Parkinson patients, parts of their brains die. Parts of their brain cells that are damaged die. So that means that we could take good cells and put them back in the patient so that the patient may recover. Now, this part here is very, very brand new things that are happening right now, but it's very, very new. So this is not something that is happening a lot, but it is happening. And the reason for that is because back last month in April of this year, it was the first patient that received the first human cells for Parkinson's disease. So this is something that is just happening right now, and we are doing the very first clinical trials. So that means that maybe in 20 years, this will become something that we can do in medicine. But for now, this is a very first breakthrough new technology happening when it comes to medicine and treatment of patients. Now, for brain, as you can imagine, the brain is so complex that for the brain, this is something very new. This is something very wild. But there are certain organs that are a bit easier to work with because we know a little bit more about them, such as the case of the heart. So when it comes to the heart is made of muscles. So the heart beats because it has muscles that allow it to beat. And we actually understand the heart and the heart cells and the muscles quite a bit better than we understand the brain. So people that have suffered from heart failure, where their muscle that allows the heart to beat has stopped functioning. So these heart cells are dying or are dead. So they are no longer working. So we in the lab, we can take cells from these patients and make them into heart cells, which they look a bit like this. So you can imagine like boom, 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 boom. So these are the heart cells moving inside our labs. And these cells then that are the heart cells that can beat by themselves, we can make them into specific devices such as this one. So this is just a membrane that we put heart cells in there. So this is what these look like. So you can see how the device moves by itself as the heart beats. And these devices then can be implanted, can be put into the heart of the patient to help the patient heart beat. And this study has been actually quite successful. The first phases of this clinical trial have already gone through, and the results show that patients that have received the transplant for these heart cells actually have an improvement of heart function, are able to have a normal life again, because these cells are helping their hearts beat and are keeping their hearts alive without the use of pacemakers. This is a big, big breakthrough. And this is exactly what you can see here. Again, all of this is very new technology. You can see these are some of the countries that are involved in using stem cells and cell therapy in clinical trials. We have brain diseases, mainly Parkinson. We have diabetes. Cancer is a big one. We have heart diseases. We have ALS is a muscle brain disease. We have eye diseases. We have bone diseases. So we have a lot of diseases that can really benefit from having a patient, using patient cells to study not only the disease, but how we can make the patient feel better, not through drugs or through replacing the cells that are no longer functioning. An interesting fact that I think I still have time to tell you is that the reason why Japan specifically has so many clinical trials using stem cells and so many different diseases. And the reason for this is really curious, because Japan is very small in comparison to the US, for example. Japan is very small, but because Japan is an island and it has a small population, the population within Japan genetically is not very different from each other. So it's very, very easy to find patients that are compatible genetically. So it's incredibly easy to just create, and they have created, a bank, so a place where they store cells that they have made. So these induced stem cells, so they collected skin cells from their population, they made stem cells, and so they have places where they store these stem cells that then they can use for all of their clinical trials. And because their population genetically is so similar to each other, it's very easy for them to just go to that place where they are storing the cells and find one that matches. In the US, for example, the population is much more varied, it's much bigger. There's a lot of people from immigrants from different places. So it's much harder to do this. That's why Japan has so many clinical trials. And this is really cool because this can make the process go much faster because it's much easier to find compatible people. So in the end, what I want you to remember from this talk is that pluripotent stem cells can create any cell type. So from a stem cell, you can get any cell type that exists in your body. And then by reprogramming, we force the skin cells or any cell in your body, we can force it to become a stem cell, which then we can use to make any cell to then study diseases and development and function. We can use these stem cells to create tiny, tiny brain-like things that we can use to study development, to study activity, to study diseases and how diseases affect development. And these stem cells are also very useful and have made huge leaps. So if you think that this first reprogramming strategy, the first stem cell created from a skin cell was made in 2012. Right now we are 2024, so it has been 12 years. In 12 years, we have these many clinical trials going on using stem cells. This is a huge achievement and is not something that you see very often. This is because stem cells really have made a huge impact in the way that we not only look for new drugs, but in the way that we can create new innovative therapies that would have been impossible just 10 years ago. So this is a very interesting field, in my opinion. But I do hope to hear more about what you think about it. And if you want to know more about this or have any questions, just let me know. Thank you.
Thank you, Liliana. Very, very interesting research that you're doing. Let me stop the recording.